Good day, I am Miguel Matienzo, and I shall be presenting my thesis entitled Production of Recombinant Giant Grouper Epinephelus Lanceolatus, Luteinizing Hormone in Dimer Form by Baculovirus Expression System. So for the introduction of my thesis, luteinizing hormone together with follicle-stimulating hormone are two gonadotropins that play a vital role in the reproduction activities controlled by the hypothalamic pituitary gonadal axis of vertebrates. They are classified as part of the heterodimeric glycoprotein hormone family, each consisting of an identical alpha and hormone-specific beta subunit that are non-covalently linked. Post-modification translations such as glycosylation and heterodimerization of gonadotropic hormones are essential for biological function. Studies were able to attest that FSH function is correlated to puberty and gameteogenesis regulation, whereas LH control gonadal maturation and spawning. Except for mammals where gonadotropin activity is clearly defined and developed, a significant aspect of fish gonadotropin functions is yet to be elucidated due to its displayed complex reproductive strategies. Moreover, the understanding of fish gonadotropins, specifically LH, mainly emphasizes on gonochoristic species. Based on previous reports in teleost, two gonadotropins were first demonstrated in salmon, wherein LH portrayed consistency in its acting role during final gamete maturation. A surge in its mRNA and protein expression level was observed, thus exhibiting stimulation of maturation inducing steroid in the effective final maturation of gonad, spermiation in male, and oocyte maturation in females. So in here, the baculovirus expression system is a eukaryotic expression system that acts as a powerful tool for producing exogenous proteins. It is widely used to produce recombinant proteins. So several recombinant proteins have been successfully generated using baculovirus expression vector system since human beta interferon was first expressed by Autographa californica multiple nucleopolyhedrovirus in 1983 due to its ability to express high yield in expression followed by a proper post-translational modification. And here, giant grouper is used as a model to study the biology of sex differentiation. The giant grouper, EPINEPHELUS lanceolatus, is a marine species classified as protogynous hermaphrodites, wherein they exhibit sex change through maturing first as females and later reversing to males. They are known to be abundant throughout the Indo-Pacific region from South Africa to the Red Sea east to Japan, mainland China, Hainan Island, Philippines, Thailand, Malaysia, Indonesia, Micronesia, and the Hawaiian and Pitcairn Island. But due to overfishing, the giant grouper has declined significantly. Moreover, the aquaculture technique is limited such that the extraction of a large amount of pituitary gland is difficult. Biotechnology was used to obtain the desired recombinant gonadotropins. So in this study, the objective is to pave way in studying the mechanism behind sex change in gonadotropin function by expressing the dimeric form of the recombinant luteinizing hormone in insect cells into baculovirus expression system. So for our methodology, the study has 4 phases: cell culture in virus, production of luteinizing hormone in insect cells, characterization of recombinant protein, and purification of recombinant protein. So for the first phase, we have the cell culture in virus. Here we have the SF21 cell culture. The SF21 cell line is an ovarian cells that are isolated from fall armyworm, where it has a cell doubling time of 18 to 24 hours. After that, we passage suspension cells. In here, they are examined under a microscope to assess the confluency as well as the general health of the cell. And to determine the virus titer, endpoint dilution is used. Endpoint dilution is a method wherein we determine the virus titer. After that, we did cell counting. So for the second phase, we have the production of luteinizing hormone in insect cells. And here, transfection of SF21 insect cells is produced by the recombinant virus in stock and to observe its performance. Virus infection takes place, and the calculated required volume of virus were infected in the cells. Another thing that was made was the fluorescence microscopy. In order to determine the success of infection, the expression of EGFP is observed under the fluorescence microscope, or the enhanced green fluorescence protein. And lastly, the determination of protein concentration was made by the use of the biotech synergy kit. As for the third phase, we did the characterization of recombinant protein. And here, Western blot is used to detect the common alpha and the LH beta subunit from among the mixture of proteins. Lastly, purification of recombinant protein was done by the use of the immobilized metal affinity chromatography, or known as IMAC, purification of polyhistidine tag proteins from a soluble protein extract. So here we have the results and discussion. So to give you the rationale behind the tricistronic baculovirus expression vector construct, previous studies on the luteinizing hormone focus mainly on gonochoristic species. However, with regards to hermaphrodites, only limited data can be obtained. It was observed that there were no direct studies associated with giant groupers, and there is a vast knowledge gap between understanding the reproductive function in endocrine regulation, particularly in protogynous hermaphrodite fish. For these species, the pituitary gonadotropins are speculated to control pubertal development and sexual transition as it matures. However, the mechanism for this has yet to be understood. Although grouper species are known to be valued at a high price in the aquaculture market, making grouper culture a growth industry, it poses a great endangerment to many of its species in the wild. A recent study has identified that LH is a factor in sexual transition in groupers. With that, the study aims to utilize the baculovirus expression vector construct developed by our laboratory for the expression of recombinant giant grouper LH, as shown on your screen. The expression vector construct for the giant grouper's luteinizing hormone, a protein dimer, consists of the LH beta subunit and the common glycoprotein alpha subunit, separated by a 2A-like sequence allowing simultaneous protein expressions. Protein tags, such as the flag tag and his tag, were added at the C-terminal of the common alpha and LH beta subunit, respectively, to facilitate purification and the protein product analysis. Since it is generally accepted that P. coronavirus-derived iris element do not work on insect cells, an alternative was used. A previous study demonstrated that the aphid infective virus, or the RHPV, contains an iris element that is fully functional in baculovirus-infected cells. In the tricistronic expression vector, a single construct containing the LH subunit and the EGFP reporter genes is used under transcriptional control of the polyhedrin promoter. Following transcription, the LH subunit is translated by a cap-dependent mechanism, while the RHPV iris-mediated mechanism regulates the EGFP expression. So after understanding the rationale behind, we did the determination of virus infection of SF21 cells. So prior to this study, 3 virus stocks were already prepared. And here we determined the virus titer, which was calculated to be at 4.42 times 10 raised to 8, 6.9 times 10 raised to 6, and 2.57 times 10 raised to 8. Due to the varying virus titer, the optimal concentration for LH expression is yet to be determined. All three samples expressed EGFP, wherein samples 2A and 2C portray high intensity compared to sample 2B. The difference in intensity may be correlated to the virus titer or environmental condition that it is exposed to. So after determining virus infection, next we have to confirm if the recombinant protein was produced and the antibody specificity. So in here, the Western blot revealed that the histidine tag LH beta and flag tag common alpha for both cytosol and media reacted with mouse anti-His antibody and anti-DDDK antibody, indicating a recombinant protein at an estimate of 16 kilodaltons and 17 kilodaltons respectively. To determine whether the antibody specifically recognizes bands, a positive control, FSH beta, and common alpha were added to test the expression's effectiveness. As shown in the figure, bands were observed at an estimate of 14 kilodaltons for FSH beta and 17 kilodaltons for common alpha, thus confirming the recombinant protein expression. A mock sample, the TNMFH medium, was added in lane 1 to determine bands that appear due to non-specific interactions. In the analysis, since the luteinizing hormone is a secretory protein, the cytosol results are given much more importance. Upon comparing the bands reflected on the blot, line 3 was observed to express strong signals and less cellular proteins for both cytosol and media as compared to the other samples. As a result, used as the basis of protein purification. So after that, we evaluated the optimal condition of infection in SF21 insect cells. In here, optimal conditions for virus infections were already tested prior to this research. However, it is vital to back up this claim. Here, 12 samples were prepared wherein it is separated equally into different day post-infections. So as shown on your screen, we have the 3 DPI, the third day post-infection. These samples were then labeled according to the MOI tested, which are 0, 0.5, 1, 2, 4, and 8. Upon observation of the expressions of EGFP, all infected samples emitted green fluorescence. Since MOI 0 contains no virus, it is expected to show no EGFP expression. Another notable change that occurred was the enlargement of cells upon infection. It can be concluded that virus replication is taking place, thus causing cells to grow in size. So here, for the next slide, we have the 4th day post-infection. So after observing its EGFP fluorescence, we identified that the vector, we know that the vector is a secreted protein. So we tested it in the Western blot. It was successfully expressed in the cytosol. The expression of LH beta was tested before proceeding to purification to ensure protein expressions. As observed in lane 7, it expressed the strongest signal than the other samples, thus concluding the optimal condition to be at MOI 1 and PPI 4. So since we already know that strong signals are expressed in lane 7, the confirmation of the formation of dimer by purification took place. So in here, biochemical experiments are widely employed in the expression and purification of recombinant proteins. An effective purification process includes the use of a peptide affinity tags, in this case, the 6 times histidine tag, which are fused to the protein of interest and used by affinity chromatography to accelerate protein purification. Upon analysis, it was observed the success of purification in the expression of the common alpha eluted from the magnetic beads E1 at 16 kilodaltons upon reacting with the anti-DDDK antibody. There were no non-specific bounded proteins observed in the wash column. So as you can see, in the elute 1, 16 kilodalton band was observed. So next, we have the expression for LH beta. It was observed to express a strong signal upon reaction with the anti-His antibody at 17 kilodaltons. Similar bands were expressed in the wash column, which may indicate that low amount of the protein of interest were released during the washed sample collection. Overall, since both subunits were expressed in the elute column of the Western blot analysis, it can be concluded that a protein dimer is formed. So to summarize or to conclude this research, the Western blot analysis for the optimal virus titer was obtained at 6.9 times 10 raised to 6 due to its high band intensity in the cytosol, as well as less non-specific binding of cellular proteins expressed. It was also noted that the primary antibodies, anti-histag, and the anti-DDDK successfully reacted with the protein histag and flag tag attached to the LH beta, approximately 17 kilodaltons, and common alpha, approximately 16 kilodaltons, respectively. The expression of EGFP facilitated by the RHIR-mediated translation emitted green fluorescence, thus confirming the success in recombinant virus infection. The optimum condition which portrayed a strong signal for LH beta expression in the cytosol are MOI1 and DPI4. And lastly, the analysis for both common alpha and LH beta exhibits clear band at roughly 16 kilodaltons and 17 kilodaltons respectively in the elute, proving a protein dimers formation. So that's it for my research, and thank you so much for listening. And I hope you learned a lot.